Greetings! For my poster today, I'll be briefly talking about how small proteins can be engineered to effectively detect diseases at early stages. The primary goal of engineering small proteins is to identify specific changes in their amino acid sequences that can lead to desired properties. As you can see from the scheme, the two primary methods for protein engineering are directed evolution and computational design. Directed evolution works on the principle of mimicking natural evolution. It generally includes four steps. The first being diversification, which generate the gene library, then expression of the protein library, followed by screening of the same for the desired function, and then finally amplifying the selected ones. Rational designs are based on comprehensive structural and functional information of the target protein. The computational methods are developed based on rational design, which are in turn based on known protein structures and sequences. But in some cases, the proteins are designed from scratch, also known as de novo protein design. The structures thereafter can be mutated in accordance to the field of application, which can vary ranging from food industry to gene therapy to remediation, pharmaceuticals, biofuels, and polymers. This is a structure of fibronectin monobody which can be used for targeting proteins like lysozyme. The areas in red denote the regions or amino acid sequences which can undergo mutation to bind to the target of interest. Each amino acid in these marked region can have 20 different combinations based on the 20 different amino acids, which implies that for mutating just 5 positions, we can generate a library of 3.2 million unique structures. The library that is generated now is screened to get proteins which bind best to the target. A very popular screening method is magnetic bead sorting, where cells are allowed to be separated by using magnetic nanoparticles coated with antibodies specific to a particular surface antigen. With this method, the cells can be separated either positively or negatively with respect to a particular antigen. The limitation of this method is that it generates low resolution binders, but that can be overcome by passing the resultants through a flow cytometer. In case of flow cytometry sorting, the instrument detects cells using parameters like cell size, morphology, and protein expression. It provides fast, objective, and quantitative recording of fluorescent signals from individual cells, as well as the physical separation of cells of interest. The binding proteins obtained after the final screening are then studied further to predict the surfaces at which they will effectively bind to the target via methods like docking. The ultimate goal of docking is the prediction of the 3D structures of macromolecular complex of interest as it would occur in a living organism. The lead variants shown here are fibronectin monobodies generated using Swiss model server that are capable to bind to lysozyme. These will bind to the lysozyme to form the lysozyme FN3 complex, which is shown here. Servers like Rosetta ZDoc will generate structures that are ranked using scoring functions to identify structures that are most likely to occur in nature. In this manner, small proteins can be engineered to bind to targets using both wet lab and computational methods. Successful binding outcomes promise the possibility to detect aggressive diseases at very early stages, even when it is beyond the scope of the prevalent diagnostic tools, thus making it a potential candidate for diagnostic as well as therapeutic purposes. I would like to thank my advisors Dr. Daniel Woldring and Dr. David Hickey for encouraging me and helping me with this poster, my lab mates from both Woldring and Hickey lab, the IQ department, Chemical Engineering and Material Science department, and the Dow Chemical Company for giving all of us this opportunity to attend the workshop and present our posters in such trying time. Thank you very much.